Hello, everybody. We're making a video about lipids and nucleic acids. Now, when we're talking about lipids, we're talking oils, butters, anything that's oil, anything that's butter, any type of butter, coconut butter, anything like that. That is going to be a lipid or a fat. So, avocado, you got your walnuts, pecans, you got the fat in the salmon, all that kind of stuff. Seeds have fat in them. So let's look at what the functions are. Now we have three different types of fats we're going to talk about. We're going to talk about steroids first. So let's look at the steroids. Let me move this video over. So steroids are these ones right here. Steroids have a bad stigma in our society because it's like, oh, sports, people doing steroids. Well, steroids are actually natural chemicals in your body, like testosterone, estrogen, progesterone, cortisol, those types of things. So insulin. So, for example, you need insulin to help maintain homeostasis in your body of your blood sugars because it takes the sugar out of your blood into your cells. And vice versa, you have glucagon that's going to take the sugar out of your cells and put them into your blood if you're getting low on blood sugar. So you need both of those hormones to help balance out your blood sugar. So one of the main functions of steroids is to help maintain homeostasis in your body. Now, another one is to help you grow, like you have human growth hormone, follicle stimulating hormone, and things like that that are going to actually stimulate the other hormones in your body to function properly, and you'll grow. So that's the two functions of steroids. Now, triglycerides are right here, and it's called a triglyceride because you have a glycerol molecule right here, and you have three fatty acid molecules right there. These are the ones that are in your food. You eat them, and they're usually stored in your body, and they're used for long-term energy storage. You get 9 calories per gram of fat that you eat, 9 calories per gram, which is pretty cool because it's double what you get from proteins and carbohydrates. Now, phospholipids right here, these are actually, you see the phosphate group right here. There's a phosphorus molecule element right there with a little bit of nitrogen. And then you got this phosphate group, and you see this glycerol as well. There's a glycerol molecule right here, and then you have fatty acids here. They actually make up the cell membranes in your cells. So they actually control what goes in and out of your cells. So they help protect your cells from infections and things like that. That's what phospholipids do. They control what goes in and out of cells. So that's lipids for you. Let's move on to nucleic acids. I'll move this over here so you can see nucleic acid. All right, so nucleic acids actually control the production of proteins in your body. That's their main function. Okay, they're still a macromolecule, and they control how your proteins are made. That's what I'm saying. So the two examples are DNA and RNA. So DNA, as you can see, is double-stranded. It actually looks like a twisted ladder. That's kind of what we say in science. This is a twisted ladder. Now, it's actually called the double helix structure. As you can see, they're connected, these nucleotides. So these are what they call nucleobases, okay? But each one of them that's connected all together, the whole molecule is called a nucleotide. So let's look at one of those really quick. So here is a whole nucleotide right there. This whole structure right here is a nucleotide. So you have a phosphate group, deoxyribose is a sugar, and then your nitrogen base, which is adenine for this one. So all these structures all DNA has that same structure, RNA has that same structure. There are just a couple differences between them. So let's look at the first difference. So DNA actually has a sugar called deoxyribose, whereas RNA has a sugar called ribose. So it's ribonucleic acid for RNA and deoxyribonucleic acid for DNA. Now look, the difference is there's no oxygen on the deoxyribose sugar here. Deoxygenated ribose. So if you think of it like that, well, that makes sense. The sugar's missing an oxygen, so it's deoxyribose. Now, let's go back here. So that's our first difference. You got deoxyribose for DNA and ribose sugar for RNA. Now, if we look at the nucleotides, the nucleobases right here, you have thiamine for DNA and you have uracil for RNA. That's one of the other differences. So RNA can actually leave the nucleus because it's not made of the nucleus. It's made from other nucleotides that are around in the nucleus, and DNA is the nucleus, so it can't leave or it would explode. So there you go. What I always call DNA, it's like the recipe book for all your proteins in your body, and RNA is the recipes that you make a copy. So you make a copy of that recipe and you can take it out and take it home with you. So it actually leaves and goes and becomes a protein. So it is the DNA is the structure that holds all the, what is it, protein, the map for your proteins, all your proteins. And then RNA is actually making a copy of specific protein structures so that it can go out and make those proteins. It's not making proteins in the DNA, in the nucleus, but it'll take it out and the proteins are made outside the nucleus. So the last one, you have the double stranded and then single stranded. That's the last difference. Okay. So they're actually pretty cool. DNA helps make proteins. Pretty awesome. So yeah, now as you can see, if we look, we have yellow, green, blue, and red. So if you notice that yellow always bonds with red, so adenine is one of the nucleotides, and it always bonds with red. That is called Chargaff's rule. It's always going to bond with red. Now, green and blue, so cytosine and guanine always bond together as well. That's part of Chargaff's rule as well. So when you're base pairing, which is you're matching up the nucleotides based on Chargaff's rule, you're base pairing. So you see that green always bonds with blue, cytosine always bonds with guanine, and adenine always bonds with thymine in DNA. Now in RNA, that's a little different because you have uracil instead of thymine. So the uracil will bond with the adenine, and the guanine still bonds with cytosine. So when you're base pairing, it's really easy. Just make the switch from DNA to RNA, just change the T to a U, and you're good to go. So all these organic molecules are essential and vital when it comes to cells and helping you function as an individual organism. So that's all I've got for you. Hope you have a great day, and I'm out.